Hello, everyone. I'm Dr. Finley. I'm kind of the course supervisor at this point. I've been asked again this semester to go ahead and put together a lecture on COVID-19. I did this last semester for the 203 labs, and it was really quite well received. So again, I'm going to go ahead and present this material for you. You're going to have a PDF, basically the copy of this, and then this will be a Zoom recording if you're not familiar with how I do these things. So all the different sections will have the same content. This was a lot to put together. I'm teaching also the clinical hematology class, so it got me up to speed. I present this information as well for them, especially I focus in more on testing and things like that, the kind of work that people will be doing. I'm putting together a lot of different disparate information, and so I also really try to keep things as up to date as possible. So this is about my bazillionth COVID lecture I put together, and every time literally things are changing day by day on this pandemic. And so I think I'm giving you the most up-to-date information. I'm also highlighting a whole series of really trusted sites that you guys can go to and try to battle against the horrid infodemic that's going on. There's a new word that's been put out. And so I learned that this weekend. So yeah, let's just kind of work our way through this and see. Hopefully this answers a lot of questions people have. It also maybe will help you understand the material that we've put together as virtual course content to really help you guys not only think about some biological questions, but also understand that the techniques and technologies and concepts that we're reviewing in 203 lab have real world applications. I think that always makes it much, much easier to digest new content and information and put it in a appropriate context. And I find things much, much easier when I can learn that way. So that's the intent. I have put in a lot of information. I kind of tend to do that. If any of you have had me for classes before, I just want to make sure that I'm as thorough and as up to date as possible. So that being said, as of today, which is February 7th, there's been over 27 million reported cases of COVID-19 in just the United States. And we're pushing a 500,000 deaths. But I think by the end of February, we'll definitely easily have passed that, probably in the next week or two. There has been some hopeful signs, but we've had hopeful signs before. So again, I think it really behooves us to be as literate and educated about this pandemic that's been plaguing our lives for a year now. And so we can get a handle on it and make it go away. That's the ultimate goal. Get our lives back. And I think information is the best place to start. So I'm going to kind of go over what COVID-19 is in terms of an infectious disease. Also the kind of testing that's out there and kind of immune responses that are important to have. So this really kind of gets into some of the PCR based kind of analysis and other things as well. And then I think it's really helpful to get as up to date on vaccinations and immune responses and what this means for us. There's a lot of noise out there right now about vaccinations. Should you get vaccinated? Can you get vaccinated? How do the vaccines work? And I think I put together hopefully some useful, helpful information. And then I kind of breaking it out into health-related symptoms, kind of some developments. There's some fascinating demographics going on, who are high-risk groups, just all kinds of diverse questions that go into this and some other related issues. And then I have here some trusted information sites that you can go to and really you know, you're perfectly capable of running your phone, your computer. You've gotten technically very savvy, having to do all of your classes virtually now. You can do this. You can research these questions yourselves. And again, especially if you go to these trusted sites, there's so much disinformation right now that I'm trying to be very, very clear. So places that you'll see, I've referenced here a lot in terms of just basic good information is presented by NPR, the radio. You can log into their online site. They have great news articles. They have worded things. They often have reviews of published information, scientific reports, and they put it in a language that the average person, I think, can understand. So again, it's really good to have that baseline kind of things, especially when you have family and friends asking questions, or you're having a discussion about how best to proceed. It's important to not necessarily have scientific reports, which are very difficult to go through, but a good distillation of that information that can be usable. I find NPR really helpful. The Centers for Disease Control and Human Health and Services federal programs yeah, are usually very good. They've suffered mightily. And I think they're starting to get themselves back, pulled back together. They've always had some decent sites, online sites that you could access, and I think very reliable information that's okay. They're getting better, is what I'm trying to say. California has got all kinds of, the state-based websites. SDSU has got an online page. The county, San Diego County has got information. You can go there. And then I find, if for specifics, Wikipedia is pretty up to date, especially on COVID disease. I mean, it's really being quite helpful, especially when I'm putting together some of these lectures, that I can just get good graphics and good information out of there that's helpful. You can just Google COVID-19, and then the other trusted sites would be, say, Johns Hopkins Medical School, Cedars-Sinai. I saw they had a really good COVID site. Obviously, San Diego County, New York Times, and LA Times, and other kinds of well-respected publications, newspapers and magazines and journals. Scientific American has had some really nice things that, if you really want to delve into it, there's all kinds of online access. I just read a paper that was out in Scientific Reports about infodemic that's going on, and defining that, and how there's just this plethora of misinformation, disinformation, and just kind of all kinds of issues. So again, this is why I'm trying to be, as this is a quickly evolving, changing situation. You need to be able to have trusted sites that you can go and find out more information. Okay. So again, it's just a huge problem. I saw that Johns Hopkins, the medicine site, had basically, what are some things that are out there in the ether that basically is basically misinformation, and how they're very careful about debunking that. I would say none of the social media based places are places to go for any kind of trusted information. Facebook, Instagram, all of these places. I think they're starting to clean up their act a bit, but it's been problematic, and I would be careful. And I think the best I can kind of say is, my family had a saying, that you may not want to be listening to that drunk barfly whispering in your ear about real good pertinent information. It's problematic, and it's kind of human nature to spread rumors, but now we can do this on a global scale, and it really has become counterproductive. Okay, so that's the first thing. See, this is some information I can pull off of the NPR site. Again, it's basically breaking out the number of cases in the United States. It's following it since about March. And when things were still at a low roar, and then this is looking at the number of cases over a rolling 7-day period. So I want you guys to kind of think about how to look at some of these graphs, so you're educated and understanding how to read what this is looking at time, this is number of cases, and this is 300,000. So I mean, we haven't been doing well since, I would say, the concern was around Thanksgiving, people were going to be traveling, and sure enough, we've had Thanksgiving, Christmas, New Year's, and it's just really starting to become problematic. I think we're on the backslide of this, but the number of deaths actually kind of lag behind. So we're still spiking. We had the huge, again, spike this past week as the biggest one-day average of people passing away from COVID-19 here in February. So this is looking like the deaths. It goes cases, hospitalization, and then deaths. There's a basically a 2 to 4 week time window that's shifting as we go. This is if you've been ill, I'm very sorry. I'm hearing a lot from students about being ill with this. If you've lost family and friends, I feel for you guys. This is not fun. This has been horrific, and we're suffering a national tragedy right now. I put up this one graph in terms of infectious cases. This was, I think, really helpful in understanding that there's, this is looking at the number of reported cases down here. So these are positive tests. This is a definite diagnosis of somebody being infected with the COVID-19 virus. And then it's looking like there's this huge pool of people that are probably infected, and they are not showing symptoms, and they have not gone in to get tested. So this is hugely problematic. They are capable of shedding live viruses, and this is where things are flying under the radar in terms of at-risk people being exposed and basically getting the disease and being very ill and dying. So it's like we've only seen, this is the tip of the iceberg here, and this is the actual iceberg that's going on. There's been a lot of questions about whether people are truly fully immunized, basically, if they've had it. It looks like people can get it more than once. And then I'll kind of break out some of the latest on the new viral strains that are out there. So I think it's a thing. The NPR site also has nationwide information broken out per state. California, you can break it out per county, and then you can break it out even further if you want to. But NPR has this really nice click on the links, and it really shows things, I think, in a very digestible way as to what's going on. Again, where the vaccination campaigns are working, which would be West Virginia and the Dakotas, who knew? Again, fairly small population size. California has been kind of struggling. And so I know I've been trying to get in to be vaccinated, and I can't get in yet. So you guys are way down on the list. So I think this is just kind of highlighting some of the useful information that you can get off of these trusted sites. They're all science-based. They're usually really as up to date as they can make them. Pretty good stuff. So that helps. When we start talking about what COVID is, it's basically a corona type of virus. There's multiple types of viruses out there in the world. There's RNA and DNA viruses. And what I'm trying to show here is highlight is that this is a novel coronavirus. It was designated coming out of 2019 year. So that's what the 19 stands for. This is looking at the genomic sequence. I'm not being snarky here. I'm just letting you know that they knew this genomic sequence back in December of 2019. So they had this in there. I pulled this up off a public database, easy peasy, nice and easy. You can do this too. And in March of 2020. And I've left it here because I want to kind of highlight the thought process behind looking for different viral strains, designing tests, and the kind of information. Once you have the genomic sequence, basically, of a virus, whether it be RNA, like the COVID-19 is, or DNA, like some of the others, you can really understand a lot of what's going on with that virus. Okay, that being said, okay, here we go. This is looking at the open reading frames that encode potential proteins for that virus. And I think it's clarified here in this illustration a bit more. So you can take that sequence and figure out what kind of potential proteins are there. And then this is a really nice illustration in that it shows the structure of the infectious viral particle. Okay, so they're not cell-based. They're basically a unique class of thing that's an infectious agent. They can only replicate if they can get into a host cell. This pertains to viral phage viruses or bacterial phage viruses. And again, all the RNA and DNA viruses, they have to get into a host cell and hijack the cell's machinery to replicate their genome, make more of their specific coat proteins, and package themselves up as brand new viral particles that are infectious. Okay, it has to happen that way. So the nice thing is that viruses are, especially the coronaviruses, are really not all that stable. So if you wash your hands with soap and water, it actually destroys this coat protein and it makes it kind of dissolve, and it's non-functional very well. Alcohol kills it, sunlight kills it. All these kinds of things disrupt this delicate structure that goes into making an infectious viral particle, and we're done with it. It can't infect you. Okay, what we're learning is that this particle is primarily being shed out of our bodies in respiratory droplets. So if you're breathing, or you're talking, or you're laughing, or you're coughing, or you're sneezing, or spitting, or whatever, you're shedding viral particles. And again, how far those little droplets go is dependent on a whole bunch of environmental conditions. So this is why we are talking about social distancing, and this is why we're finding that mask wearing is hugely helpful. There's been a bunch of disinformation out there, especially in the United States, that this doesn't do anything, and that's not true, period. Okay, so I'm here to dispel that. So understanding this is really important. So if you're shedding virus, but you got a mask on, you can't infect other people, okay? If you're wearing a mask and somebody is shedding viral particles, you're protected, at least in part. Okay, if somebody's sick, they should be staying home and not being around other people unless they get desperately ill and need help. I mean, there's just all kinds of common sense things that if we had pulled together and gotten with the program, we maybe would be done with this thing. As it is, we're still in a third wave, and the deaths have not started to come down yet. So you see where I'm going with this? I mean, it's just common sense kind of stuff if you understand viruses and what they're all about. So again, this is highlighting that there's usually a part of the lipid membrane that's coming from the host cell, and then there's all kinds of viral proteins that are essential to assemble this viral particle, and then in the center is the RNA genome, or in some cases, different viruses, it's a DNA genome. This is kind of highlighting how they have to get in. And so you've seen probably pictures of the coronavirus. It's got these little spiky protein little knobs sticking off. This interacts with a receptor type on host cells. And this has evolved now to be fairly specific for this ACE2, and basically that's a cleavage event, but this the TMR PMRSS receptor on cell surface. So the epithelial cells in the lining of the lung and the nasal passages has this receptor, they can become infected, and it's located other places, okay? So if the cell is expressing this kind of interesting complex, this results in the virus being pulled into the cell. It basically releases its genetic material, and then it replicates that and then starts repackaging itself and then shedding virus. That's what we're talking about. So this is hijacking that cellular machinery to make these specific proteins, and now it's ready to these new particles are ready to infect again. Okay, this is kind of a basic virology 101 here. Okay, so hopefully that makes sense. How do we test for this? So I showed you the genomic sequence, and this has been the baseline test that everybody has, if you've had the Q-tip up your nostrils and tested, then this is what you're doing. You've all heard of PCR, I hope. The fact that you can have amplification of DNA if you know the sequence, and you can generate a set of primers to that sequence. You can amplify a DNA fragment of a particular size. There's some, you guys are going to learn about this in detail. If you don't know about it before, your TA is going to be going through all of this with you. And so this is basically a gel where we ran out the PCR products. This is a ladder. We stain this with a compound called cyber green that gets into the DNA that you've amplified. And you can actually visualize bands, make sure that they're the right size. So this is looking at a ladder. I think this is 1,000. So this looks like about 1.2 kilobases in size. So that'd be 1,200 bases in size. Smaller bands, things like that. You can see the wells here, okay? And so you can see relative size, because they all run at a certain, bigger runs slower than smaller, and that's what the ladder is showing here. And then you can kind of get intensity. So relative amounts can be figured out this way. So here is a band, obviously is lighter than this one. Okay, so there's more DNA here. It's qualitative, not really quantitative, but you could quantitate that. If you want to quantitate something and you want something that's extremely sensitive, we have this other kinds of testing material. And I didn't know I have clickers here. Here we go. And this is looking at a molecular diagnostic PCR-based test. This is called real-time quantitative PCR. So what this is basically a PCR reaction. You have a DNA template, starting material. You have primer sets, and you run, in this case, we're running 40 cycles fairly quickly. And this is actually, you have cyber green that's in the individual reaction mixes. And as it's cycling, you're starting to detect the fluorescent glow, okay? And so you'd usually have a pretty specific cutoff where you're starting to pick up the cycles. Usually it's right around here. If you get up here, it kind of becomes non-linear, but you can see that this one is coming up quickly after like 18 cycles. You're starting to detect it. Here's one that's, coming up really quite late. It meets this particular threshold, and that would be at about 27.5 cycles. So more DNA start in the starting reaction, less DNA in the starting reaction. We do these kinds of things actually to look at levels of gene expression. So we're looking at mRNAs. You can do this for viral particles as well. Okay, you can use this for bacterial infections. You can use it for RNA viral infections, and you can use this for DNA viral infections. This works great. And so again, basically you need a biological sample. You need to have the appropriate Taq polymerase, and you guys are going to be learning about that. You need the cyber green in the reaction mix, nucleotides, and then specific primers that you've gotten from knowing the genomic sequence of the COVID-19 viruses. So back in, what was it, February of last year, about a year ago, when the US was like flailing around because it didn't have reliable COVID tests. I mean, I'm sitting there going, I could design the primers and run it myself. I mean, this is not rocket science at this point. There was a really researcher in Germany. He had back in, I think, 2019, at the very end of the year, heard about that, got the sequence, designed some primers, and then was shipping off his reactions and his primers to, I think it was Hong Kong, and they beta tested it for him, said, works great. So the World Health Organization had these kits ready to go, and the CDC said, no, we'll develop our own, and then something got screwed up. So we've been late in the game in terms of testing, period. And so hopefully this makes sense. I mean, this is something, if Dr. Finley could do this and understand the technology, everyone else can too. And so why this failed, yeah, let's not go back there. Let's just, start moving forward. We got it fixed. We can do testing now. Again, we haven't been able to scale up. There's a limit of reagents. I've been trying to buy Cyber Green for this stuff, and there's delays, and it's just the demand is so huge that all these other technical things starts to pop up. So it's been very interesting. So you can do a saliva-based test. I mean, it's just, that you can, this is really scalable. If you can work on together scaling, okay? So again, I hate to belabor these points. This is just looking at the equipment. So this is something in my lab. This is the quantitative RT-PCR piece of equipment. You can see the little things here. We're looking at RNA expression levels. We have a 96 well format, and should be pretty straightforward. This has got a really high sensitivity detection system. So it's right here. So there's, each little well has basically a detection camera system hooked up to that. Here's where the PCR happens. This is the thermal cycler part. And again, there's photons of light that basically excite whatever's going on into this plate. And then it detects it. Okay, and it does it for as each cycle is going, as the amount of DNA is going up, you're picking up the cyber green, and that's why you get these nice amplification curves. Okay, and then there's some software, and we have some ways of analyzing and getting relative amounts of things. Okay, you can have the 96 well format, and there's also, I think it's a 384 well plate. These are hard to load. The 96 is bad enough, but oh, and we're doing this. Okay, so this is very odd. I didn't know I had the that kind of a thing. So that's just kind of highlighting all of that. Hopefully that makes good sense. And come on, you. So this can be scaled up. There's commercial hospital diagnostic lab grade equipment. They're running the 384 format. So samples come in from people, they're sent to the lab prepares it. Again, if it's an RNA virus, you have to convert the RNA to DNA. And then so it's RNA, it has to be turned into DNA. Again, specific DNA probes that hybridize specifically with a sequence are used in there. So they're targeted probes are used to do this. And then you can get this amplification figured out. So you can get almost relative amounts of viral titers. And so you maybe have somebody that's barely just got fairly infected. They probably aren't showing symptoms as of yet, but again, they could be shedding virus, and they unknown. Some people have their whole disease course where they're shedding virus, they're contagious, but they don't have any symptoms. This disease has been running the gamut where people just get deathly ill, hospitalized, and do not survive it. And people that are running around don't have a clue that they even have it. They don't have a sniffles. Nothing's going on with them. So it's, and then everything in between. This is how hard it's been to figure out what's going on. I'm just showing you the CDC primer test kit. It comes in little packages. So again, you can, hospitals and diagnostic labs use this technology routinely to test for a whole range of pathogens. There's a company here in San Diego, Hologic, they're doing this right now. They're doing COVID, but before then they were really specializing in a lot of the venereal diseases and knowing exactly which strain of, whatever is out there. So again, malaria, all kinds of things can be tested this way. It's a PCR based. So all you need is a bit of information about the genomic sequence. And then you're off to the races. You can design this test. And it be exquisitely specific. Okay. Okay. There's other kinds of tests, and I think this is worth breaking it out for everyone. You can look at active viral infections in terms of looking for those coat proteins. So again, it's important to understand what that's all about. So again, it's an antigen-based test for viral coat proteins. It's basically saying you have to have some of the proteins for basically it's looking at the active infection would be, looking for coat proteins. So you need to be able to have antibodies that are directed at this. I've generated my own antibodies. There's stuff called monoclonal antibodies that can be used to design these tests. They're relatively straightforward to design and get up and running. It takes time to produce novel antibodies, but once you have that up and going, then you can really have you could scale it, and again have basically tests for the actual viral particle proteins. Okay, not just the DNA, the viral particle. And that's a pretty good indication. If people have got the proteins floating around, that means they're actively shedding virus again. Okay. They're working on a rapid diagnostic test. Again, this is similar to pregnancy tests. It's again antibody based, and I thought I'd point out that these kind of rapid tests are being used in remote places to check for malaria. You could have a HIV test sent to your home and get pretty accurate results. There is some false negatives and some false positives, but again, it gives you a kind of a clear indication usually about what's going on. Usually the PCR based is much more, it's really pretty accurate. But again, it might be missing certain things. So this has its applications. You don't have to have fancy schmancy equipment. And again, I think these things are pretty stable, so they can go into remote areas like a pregnancy kit and work quite effectively. So again, it could be all kinds of biological samples, saliva, nasal swabs, pregnancy tests. You basically pee on a stick and it comes up positive or negative. You get a positive thing here. So this is almost in the works where you could order one of these COVID tests, have it sent to your homes. I don't think they're quite ready yet, but these are being worked out. Okay. And then the other important thing to understand in terms of disease and things like that is if your immune system has responded to a viral infection. So this is looking at, it's kind of not necessarily a test of active infection, but it kind of gives you a sense of, have you been exposed? Have you developed an immune response to the virus? And have you cleared the viral infection? So you can have these positive antibodies and not have any more virus. You've cleared the virus and you're no longer contagious. Okay. So this is important to understand. That it kind of gives people, the medical field a sense of, okay, you were sick here. you had the virus, we had a positive COVID test virus, you've been sick, and now you're better. We can check for virus, but again, by PCR, and we can see if what your antibody titers are. This kind of indicates how well your body responded and how long you're probably resistant to infection. Brand new virus. We got no clue how people are going to respond. It's been around for a year. We're still learning as we go here. So this is important to understand. This is fairly straightforward. Kind of assays. And again, some of it is enzyme linked immunoabsorbent assays. So this would be kind of ELISA sort of thing, where you have the viral protein again, and your antibodies would bind to it. And you could have a whole panel of different kinds of proteins from the virus and seeing how many different antibodies that are able to cross-react with different kinds of aspects of the virus. That also kind of goes into antibody design. So if people are mounting a huge response to the spiky thing versus something else, then you know which one is probably going to be a good target for the development of vaccines. Okay. So this is looking, and again, this is fairly straightforward, everyday kind of assays. This is kind of a colorimetric plate. This is for HIV detection. We have a 96 well plate format. You can get a 384 well plate format, a colorimetric assay. You put it in the plate reader, and it looks at the development of a color. Easy peasy, nice and easy, used routinely. I have access to this too. So it's really, again, something that is very common. Hopefully someday you guys will, in a lab course, be able to do these kinds of assays routinely and have it as not a huge mystery. This is fairly straightforward to be used. Okay. So this kind of is my segue into, what am I doing here? How do we develop vaccines? So the vaccines are starting to roll out. There's lots and lots of numbers being discussed. I think I'm hearing a lot about people being worried about this, that, and the other thing. And I think these are all, really good, valid concerns. What isn't been so good is the level of misinformation that's out there again, and people not having trusted sites that they can go to learn more. And again, I'm all about having everybody learn more. So when you're talking about developing a vaccine, you usually are working with some part of the pathogen, whether it be viral or bacterial or fungal or this, that, or the other thing, like a little parasite like malaria. So, and so you can do that a lot by just looking at sequence, understanding what their protein profiles will look like, and then going for the thing that would be the best in terms of blocking future infections. Okay, so 3 main things that you need to have to develop a proper vaccine. You need to have something that elicits an immune response. You're usually wanting to test initially the production of antibodies. That would be a primary response. That's part of the acquired immune system. There's another part too that seems to be playing a huge role in COVID-19, but I'll touch on that in a sec. So again, this is looking at what's called an epitope. That's part of a protein that's specific for the pathogen, that you have very specific antibodies pointed right at it, and that will bind to it and elicit an appropriate immune response. This is fairly easy to do. I've had things where antibodies are great research tools. I developed my own. I know how to do this. What's different and what I've not really worked on is the next part is sterilizing immunity. Once you have a vaccine, it's eliciting an immune response. Does that block the virus from ever replicating? So again, if it's binding to those bumpy proteins that bind the receptor, that would block it being able to infect a cell. So that's kind of, that takes time to evaluate. but that's looking pretty good for most of the vaccines that have come on board and the ones that they're still in the process of testing. Then the big question, this is where everybody is very concerned, and is in terms of safety. So, it's great. It elicits immune response. Oh, it's even better. It seems to be really preventing disease. Now the question becomes, where's the sticking point? And people, I think, are rightly concerned about this. But I think from what I've been reading, the things that have been approved so far, the different vaccines so far in this country and then in Europe and other places, looks like they've met all of these criteria, that people do get a sore arm. There have been a couple, few people that have had a much more severe reaction, but otherwise, a sore arm you can live with. That actually is saying that your body is responding in an immune kind of way to the vaccination. I always consider that a good thing. So I've gotten lots and lots of influenza vaccine. I get that every year. I get a little bit of a sore arm and that's it. I'm fine. And I have not had the flu for a long, long time now. Yay. And so again, I think we've become much more sophisticated in our understanding of potential adverse side effects. And I think these have been taken into consideration with the new design of the COVID vaccines. Okay. So again, this is part of the medical research plan for developing vaccines. They if you can't pass all three of these, then they are not put on the market. People, the general public it's not given to them. They have to pass all three of these kinds of things. I'm finding some really nice information now. It was a little bit sparse, but this is, I think, out of Boston University. Their website, the med school there is been putting up some really nice information. I thought it would be kind of nice for you guys to kind of look at the different cellular components involved in eliciting an immune response. And the one that we're focusing in on right now is the production of pathogen-specific antibodies. There's also a cellular mediated immunity too. So this involves cytotoxic killer T cells that go in and glom onto infected cells and eliminate them, that they have a virus in there. I think from what I've been reading is that this is great, but this also helps the production of antibodies. This also helps the directing killer T cells as well. Okay, so this is the cell-mediated immunity. There's these parallel systems working together to keep a sterilizing immunity going on. How long this lasts is very important. Sometimes this is why you need vaccination boosts for, this, that, and the other thing. It's pretty common. And again, this is a really nice breakout of all the kind of different vaccination platforms that are out there. You guys don't need to memorize all of this, but we've got quite a few coming on board. Right now, we have the RNA vaccine. So basically, they have a piece of RNA that encodes for, say, that Nabi protein. They put it in a secret lipid. They make like a salad dressing, an emulsion of lipids. They package it in there. The immune cells gobble it up. They produce that coat protein of the virus, and then they present it. And this kicks off this whole immune response. So you're never having the virus in there. An infectious viral particle, it's not in there. It's just a small piece of messenger RNA that encodes the protein. It's extremely elegant and it seems to be working really nice. So what's the difference is the magic salad dressing that they package it in. So right now we've got a Moderna and a Pfizer vaccine that are both mRNA based. There's some other ones that are coming up. I think Johnson & Johnson and AstraZeneca in Europe has some different kind of mechanized things coming up where you have an adenovirus vector that's basically has another fragment of the same sequence that it infects the immune cells. They produce this protein, and you're off to the races again. It's the same kind of thing where you're getting targeted things. The old-fashioned way was to basically have, say, an attenuated virus. This is how we got rid of smallpox. You have an inactivated killed virus. That was how the original polio vaccine was developed by Dr. Salk here. He was in Pittsburgh when he did it, but he's got the Salk Institute here, even after he's passed away. So we've got, decades and decades, and hundreds of years actually with smallpox in terms of understanding how to vaccinate people. Okay. And then there's all kinds of little protein subunits and yada, yada, yada. There's all kinds of this nice stuff. And again, this is another illustration because this is up and coming. I'm trying to make sure that I understand how these vaccines are working. I'm also trying to evaluate, effectiveness and safety, and report back to you guys. So this is kind of the DNA or RNA virus, RNA vaccine, live attenuated, and they're being developed in, I think India's got it coming up. You've got inactivated, so you basically grow up a bunch of the COVID-19 virus and do something horrible to it, and then that's no longer infectious. Remember, I said they weren't terribly stable, and then you can inject that. That's how polio was dealt with. You can produce artificially subunits of those proteins in the lab and purify those and inject those. There's Novavax and stuff like that. And then you've got the viral vector. So that's AstraZeneca. They partnered with Oxford University in Cambridge, and Johnson & Johnson's is coming up. So again, there's different kinds of things. This was developed for Ebola. This is, say, the active attenuated is measles, mumps, and rubella, the MMR shot that kids get, and also for chickenpox. This elicits, it's not a very happy virus. You inject it, you mount immune response, and then boom, you clear it. And then they usually are pretty effective and long lasting. So I think having these conversations, you guys are in the front lines helping family and friends. I think this is important to have this information. And if you want to find out more, there's, again, really great trusted sites to kind of figure this out. One of the things I've been hearing is that once, when they talk about how efficacious a vaccine is, if it's been delivered properly, and they say it's 94% effective, what that means, it's not that you will never get COVID-19 virus. It means that it's efficacious in preventing symptomatic coronavirus disease. So again, that means that people are not getting so sick that they need to be hospitalized, and nobody has died if they have gotten COVID-19. That this is to be clear as to what we're talking about in terms of being, it isn't complete sterilizing immunity. It still has a huge benefit. It keeps people from, it helps them clear the virus. If they get the start of an infection, they don't get as sick, and they don't land in the hospital and pass away from this. So this is a huge step forward. It doesn't mean that somebody can't get infected and still be shedding virus. So there's some nuances in understanding what vaccinations mean that I hopefully it's becoming crystal clear for everyone. It doesn't mean that you will never get it. It doesn't mean that you can't spread it. We still have to do things like wear masks until this is not rampant like it is right now. I mean, they'll let us know when we can stop wearing masks. But again, this is the first giant step forward to getting a handle on this and getting our lives back. Okay. So again, this is, I think, as clear as I can make it. So again, Pfizer and Moderna aren't the only ones. There's things being developed everywhere. I mean, I think China has got something coming up. They have such a low incidence of disease. They're actually testing it in Brazil. India's got some things. Russia's got some vaccinations coming up. And obviously Great Britain, you've got a whole series of things with AstraZeneca. It's based out of Great Britain and Sweden, that biotech company. Okay. Hopefully that makes, I'm trying to make this as condensed and sensible as I can. Hopefully this is not going too long. So again, there's some really important information I think needs to be relayed in terms of the new variants that are out there. And again, when you have millions of people infected with a virus and having problems with this, the more virus replicates, the more any kind of a microbe replicates, the more you have an increased likelihood of them having mutations. And then if that mutation has a selective advantage, then you're off to the races. And this is exactly what we're seeing. I knew this was going to happen. I think most people in the sciences understood this was going to happen, and now it's starting to happen and pop up. And so definitely things, there's a variance in the United Kingdom, South Africa, and Brazil. Again, pretty active infections in both places. And again, they're picking up point mutations that again, if it's changing that Navi coat protein, it maybe binds the human receptor even better. And so it makes it more infectious. It can also make a virus more virulent, and it can also kind of help, kind of get it underneath the immune system. So there's multiple things that these different mutations can do that can cause problems. Okay, so how do we test for that? And this is how they first figured out the genomic sequence of the original starting COVID-19 virus. And this is called next generation sequencing. I'm not going to get into it a whole bunch. If you guys are interested, you can go ahead and look this up on Wikipedia. They've got a nice page on that, and they can give you a lot of details, but you don't need primers. You just sequence the DNA and then do alignments with the sequence to figure out what you got. So that's really hugely helpful. They're doing this a lot right now. And so there's some links here. This is an NPR article that I found. It's kind of, the title is What's Going On with the Coronavirus Variants, an illustrated guide. You can access that link, read that article if you're interested. I think it, again, is helpful. Okay. I think it's also helpful if you guys know that there has been some advances in terms of treating people with COVID-19. Again, it requires access to adequate medical systems, and not all places have that, including places here in the United States. So, but that being said, there's a broad spectrum antiviral that was first developed to fight Ebola. The FDA came out with an emergency ruling to go ahead and use it for COVID. It's proven to kind of shorten this and decrease the severity of the disease. So that's helpful. There's some anti-inflammatory drugs. Dexamethasone is one of them. It's a corticosteroid. One of the hallmark features of this infection is kind of severe inflammation. And this helps kind of calm that all down. And then there's also some monoclonal antibody treatments. So again, you grow up a whole bunch of these antibodies in a particular kind of way. You inject these antibodies via an IV transfusion. And again, they basically go to town attacking the virus and getting the patient's own immune system to go, oh, over here. Let's go deal with this. And again, these are the brand names that are out there. Again, this has been approved by the FDA. And as I understand it, President Trump received all 3 of these treatments when he was hospitalized with COVID-19. And he made a very quick and remarkable recovery because he had access to state-of-the-art treatments. One of the things that not everybody has access to, I mean, if people having, again, disease that's not responding to this kind of stuff, or they don't have access to it. Very ill patients, they find that laying them flat and prone really helps them breathe better. And so this, I pulled up some videos from WebMD and YouTube, kind of illustrating, if you're wondering why the medical profession is exhausted, watch what a team of 5 to 6 people have to do to turn somebody that's been in a medically induced coma, is on a ventilator, has all kinds of things, hooked up to them. How do you flip somebody that's unconscious several times a day? I mean, and they have to do it in a very trained, organized way, and working as a team to get somebody properly turned. When I watched those videos, I went, oh, now I understand why everyone is so tired and exhausted. They're just worn out. I mean, they're working really hard all the time, trying to help save people. One of the big disinformations that was out there that's proven to be not helpful in any way whatsoever, probably causes problem, is this hydroxychloroquine that was pushed about a year ago. So that's not useful. Don't even go there. And then there's, I think, some really other interesting things that are out there in terms of an extraordinary patient that they have a new article out. I put up the New England Journal of Medicine link, but then NPR had a really nice summary of it, if you guys want to look at that. So you had somebody that was immunocompromised, got COVID, and he hung in there for a long time. He was, there was Herculean efforts to try to save this fellow. He did pass away from it, but they did this next gen sequencing with him throughout this, and they could watch the mutations start to happen. And so this was fascinating stuff to me. I think, if you're interested, please feel free. But we're learning so much, and we're starting to employ the technology that we have and our huge knowledge base to understand that even better. There's also a book I have not read that I think is potentially interesting if you're into the more the socioeconomic aspects of what's going on, is the Epidemic Illusions. Again, I have not read this, but I think this is something I'd like to get. And when I get a minute, give it a look in terms of why do we have such inadequate healthcare around the world, and how could we, much more effectively deal with pandemics that pop up. So you get the COVID-19 vaccine. I kind of broke this out in terms of what you can and cannot do. Basically, I think it kind of talks about all the things that I've already kind of brought up. That is it 100% protection? No. Is everyone different? Everybody responds, their immune response is different and unique. A moving target, we're picking up variants, right? So hopefully the vaccines will still work. There's ways to, do an in-depth analysis of that, and then maybe tweak these vaccines as we go. That I think is being considered right now. I mean, get ahead of this thing. So that needs, I don't know how much additional testing needs to be done to accommodate that, but it's a thought. And again, this is talking about being a silent spreader. And then how long will your immunity last? Is it a lifetime? Probably not. Is it, 10 years, 5 years, 1 year, 1 month? I mean, we still, again, brand new disease, we don't know. And again, everyone is different. So we're still trying to figure that out, get proper tests on board so we can, just grind through this and figure it out. I think that would be important. I think, go back. There's some interesting information about gender-based differences with COVID-19. We're clear about the age. It seems the older you are, the less well you respond to this thing. The younger you are, the better you handle it. Not always, but kind of. There seems to be very distinct racial demographics that are tragic and problematic to deal with. But then there's this huge big thing about gender. And again, I'm not hearing a lot about that in the news, but as soon as this popped up in China, they saw immediately that men did not handle this as well as women. They got sicker and they succumbed to it at a much higher rate. And I think it's like a 40% increase for men versus women. And so that's huge. So guys, don't get cocky. You're kind of sort of an at-risk population. And so, and I don't think that's been really discussed very much in kind of the general press. Why this is, I don't think guys are less clean. There's supposedly you guys don't wash your hands as much, but I kind of think it's much more of a biological based question that the immune systems of men and women are different. And so what women have problems with is different than what men have problems with. So again, when you talk about these heightened inflammatory responses, I think guys, you're a little bit more prone to having issues with that, and that can push somebody into being much sicker and having to be hospitalized. Okay, so I think that's kind of what that's bringing out, and I think it's something to be aware of and consider. There's lots of work being done around the world. This is a pandemic. That means it's affecting everybody everywhere. And I kind of follow a bit more of what's going on in, say, Scandinavia. I have friends in Norway, and they were commenting about Sweden going with, yeah, let's not worry about it so much, and we'll get herd immunity going. That was a kind of a popular concept here. And so there's been kind of basically Sweden has been kind of a guinea pig for all of this. And this is kind of breaking out that report. I pulled this down off of WebMD, and it don't look so good. They're not, I think they're abandoning this idea. It's clear with neighboring Scandinavian countries that did go into lockdown, that did, insist on mask mandates and things like that. They're not doing anywhere near as well. They have much more disease. They have much, higher incidence of hospitalization and death than all of these other countries. So again, I think there's value there on understanding that, we can learn from other places. There's just a recent thing that came out this week about India. And I just was like, oh, when this hits India, it's going to be horrible. And it has been. And they're, it's a very populous country. It's a very diverse country. I mean, the socioeconomic strata is huge. And so, yeah, it went through India like wildfire, and it's still there. But what they did partway through that was institute a really stringent mask mandate. So they're starting to open up, but if you're outside of your house, you have to have a mask on. And so they're finding people and then handing them a mask. So the police are actively all over this. And what at the peak, they were getting 100,000 infections a day, which is huge, but it makes sense. And now they're down to 11,000 per day. So it has been effective. And again, I think if we got serious about wearing masks for even 100 days, we could, knock this down. So it's not just vaccinations. It's not just having great testing. It's a whole kind of holistic approach to dealing with this pandemic. So again, this is a link to this article that again was in NPR, very easy to read, very digestible kind of information if you're interested in more. India is also racing to produce its own vaccine. And I think they're still in the process of testing it, but they have a great research community. And yeah, I think hopefully they're going to have something really valuable they need to go for volume and cheap. And so I think they're well on their way to producing something very effective. there's a couple little bright spots here. I thought I'd throw this out. The big concern was for women that are pregnant. I mean, they're always considered an at-risk group. And there has been a few issues from that, but again, they're hanging in there. Pregnant women are hanging in there. And the huge thing is that it looks like there's minimal impact on the fetus. If mom gets COVID, usually the babies are okay. That was huge. And I don't know if you guys remember the whole Zika viral outbreak that happened in South and Central and America and the Caribbean islands. It's still there. It's still a problem. But again, that had profound effects on the development of babies. So we're not seeing that with COVID. That looks so far, yay. One good thing, huh? And then I'm really kind of a proponent on the development, distribution, and access of vaccines worldwide. Until we get a handle on this, with global travel and stuff, if we don't get the whole world get this under control, then it's going to be a problem. And this is just kind of breaking out. I think this was the end of last year. I kind of pulled this information up. It may be better now. I mean, it's moving quick, guys. But US is basically purchasing a lot. The EU, India has a lot, just the United Kingdom. They're really wanting to get lots of vaccines. When you start to go into other places in the world, it really trails off in terms of what was potentially their ability to get vaccinations. So again, with some of the stuff coming out of China and India, they're really going for quantity and stability of the vaccine product that they're working on. So I'm hopeful that that's going to be really effective. And so again, there's all kinds of, we had COVID misinformation, and now we've got vaccination information. It's a thing, and it's going to have to be dealt with. But I think hopefully that makes good sense. And I'm going to end here and just reiterate that we're not out of the woods yet. We know a lot more than we did a year ago. We're getting our collective acts together to deal with this. That's great. I think if we can pull together as a country and be okay with dealing with a bit more disruption in our lives, wearing masks, socially distance, and nut it out a bit more, we'll be okay. And people getting vaccinated. But again, until then, we need to wear masks, socially distance, with the things we've been doing, and wash our hands. And then hopefully get this under control, get our lives back. And then if you guys are interested, there's a whole lot of misunderstanding and disinformation about vaccinations. I used to teach the 366 lecture class, and I had a whole lecture worked out on that. If people are interested in this, I'd be happy to post all of that information as well. I mainly kind of focus in on vaccination and autism, and how that urban legend got started, and how it does not play a role in this complex neurological disorder. So I'm just putting up fun pictures of one of the last little girls to get smallpox. There's some really good information resurfacing on the last time there was smallpox in New York City in 1947, and what the city did to deal with it. It was just amazing. There's some YouTube videos if you guys are interested. I can send that out. This is a little kid with measles, and then this is looking at polio and the type of basically neurological issues and limb deformities that came from that, and highlighting an iron lung. I think what has not been happening for you guys is realizing that each generation has to understand infectious disease and the methods that we use to combat that. And then once that, every time you guys, you aren't born knowing this stuff. So, I think it's important for these kind of conversations to be had. If not, then we're left with, the nonsense that vaccinations cause autism. Okay. And once you've eroded the public trust, it is really, really difficult to get it back. And that, I think I'll stop there. And hopefully this resonates. Hopefully you find this helpful, and stay safe out there. Dr. Finley, over and out.